Welcome to my poster about network modeling of inflammatory mediators in osteoarthritis. Diagnosis of osteoarthritis is based on its clinical features, and the most representative are synovitis, bone cyst, and cartilage destruction. Its management only deals with symptoms as it's based on painkillers, and finally, it might require total knee arthroplasty. Mechanisms are not fully understood, but we know that they affect the whole joint. Also, chondrocyte, the only cell type of cartilage, become a catabolic cell. In fact, degrading events are higher than the anabolic ones. We know that the catabolic response is driven by inflammatory mediators, but interactions are by no means intuitive. This and the fact that there are a lot of information from multidisciplinary areas makes it difficult to integrate biological knowledge. That is why we will apply a network-based model as a computational approach in order to understand inflammatory mediators' connection to chondrocyte metabolism. Information about inflammatory mediators at tissue level is well known, and also how they affect on cell metabolism. But understanding the connections between them is not a straightforward information, and that is why the main objective of this work is to find a semi-quantitative regulatory network that can control chondrocyte metabolism by obtaining the dynamical information of it. Network-based models is a powerful framework to study signaling routes within a cell. But first, a static graph must be developed. There are different strategies used to construct, but we used a knowledge-driven approach by combining it through an optimization step against experimental data. In order to obtain dynamic behaviors, the static graph is translated into a set of ordinary differential equations based on network topology. The system, when it's solved numerically, it reaches a steady state, which can be extrapolated to cell state or cell phenotype. Indeed, these steady states can change according to the imposed initial conditions. Then they become a very useful tool to explain dynamic cell behavior under different conditions, and that is why they are useful for calibration and validation purposes. The detailed methodology is explained here. First, a static graph is designed from literature. In fact, we created 62 peer-reviewed articles to develop a first literature-based static graph. It only contains information of soluble mediators and interactions on chondrocyte metabolism in terms of structural proteins and degrading enzymes. It does not contain intracellular mediators such as transcription factors, and this will help us to compare our theoretical data to experimental data. After that, an enriched network is constructed with the help of string. String finds edges automatically within curated databases and text mining. There are more options, but we only are interested on these two. Then we have introduced missing links from general knowledge in an enriched based network. Finally, network topology is optimized against experimental data using a genetic algorithm. The fitness function computes an error between experimental data, which come from cultured chondrocytes, and our theoretical data, which come from steady states with different imposed initial conditions. Finally, the model is assessed, first quantitatively and then qualitatively. The model is evaluated by its ability to reproduce chondrocyte activity based on reported actions in the qualitative evaluation, and then we test the model independently against a total independent experimental data. So if we move to the results section, we can see here the three networks developed. I would like to show you 6 types of steady state 2 for each type of network. If we take a look at the literature-based network, we can see that in a basal situation or when no initial conditions are imposed, our network goes into a catabolic state. But then, when it is enriched, our network per se goes into a healthy state because it expresses anabolic nodes. Then it can integrate a catabolic stimuli with significant difference. and the same happens in the optimized network. However, here in the baseline, AL4 is not expressed. This expression is associated with mechanotransduction pathways, and this point is discussed in my poster presentation about integrative modeling of chondrocyte mechanotransduction and biochemical complexity. The independent validation shows us that when we plot the normalized root squared error between our theoretical data and a total independent experimental set, our model could represent almost the 81% of the responses tested. If we take a look at the qualitative evaluation, we can see that the optimized network is the best in representing qualitative outcomes extracted from chondrocyte behaviors in literature. We also test a treatment in an hypothetic osteoarthritic knee. To that, we stimulate our model with the main pro-inflammatory mediators and then with the molecules present in an autologous conditioned serum. When these conditions are reproduced, we can see that our model can reduce effectively pain mediators and degrading enzymes, but structural proteins cannot be fully re-expressed. As this might not be good enough, in fact, it represents the real cases. So as main conclusions, we can say that the literature-based network is catabolic deviated because osteoarthritic information also is. Then network enrichment helps integrating metabolic switches between catabolic and anabolic states. Then the optimization process helps us reproducing almost the 81% of the responses in a total independent experimental set. And finally, our optimized network helps explaining chondrocyte reported actions as well as reproducing a real case treatment.